So, Jamie, you're going to tell us all about the genetic sequencing of coral work you've been doing over the last couple of weeks.
What I thought I'd talk about is the metabolomics work. So we've just run an initial pilot experiment. We're looking at two species, Acropora meliopora and Acropora hyacinthus. And we've got colonies of hyacinthus from Singapore and Australia. And basically what we did in the pilot was to take fragments and remove apical polyps, so the polyp at the end of the tip, and then radial polyps from multiple branches, from multiple colonies, from two species. And we snap froze them in liquid nitrogen. And they've gone up to a university, and we're going to be looking at processing those to look at the metabolomic signature. So ultimately, metabolomics, you have your genome. Your genome basically it's kind of like the recipe, the recipe of what makes you, but then the sort of ingredients of it's the ingredients of what makes you, and then when you mix all that together, you get like a soup or something. They're kind of your proteins, and then I don't know, and then the metabolites as a result of that is what your body's then done with those proteins. So it's the kind of finest level of genomics work that can be done at the moment. So
you're cataloging. You're basically starting to freeze the DNA of the specific species for reference. You were saying that you're taking them from the growth tips, as well as from a cross section of the coral in terms of the cells you're freezing. Is there a reason? What's the reason?
Well, at the moment, we just want to find out, is there going to be a difference between the apical growth point, the apical polyp, and the radial polyp, and whether there's a difference within a species, so within individuals of a species, and then also if there's a difference between species. So, we took like 50 odd samples, and at the moment we have no idea. There'll be, no doubt it will come back way more complicated, but then that's the starting point. Then we're going to try and figure out what the question is moving forward. So this is like phase one of the project. We've got phase two. Once we've got that data and we've sort of hopefully got a paper out of it, we'll then be looking at using, what we want to start looking at is resilience. So we'll do thermal stress experiments. The theory is that our Singapore brood stock is going to be more resilient, just because the environment in Singapore is warmer than Australia. So if we do thermal stress experiments with the parents, what we're likely to see is bleaching response from the Australian corals, but no bleaching in the Singapore. And then can we understand that at a metabolomic level? And then taking that another step further, which is what we tried to do in December during the spawn, is then mix and match the species, both within species and between species.
Because presumably the warmer water tolerant corals have a different genetic composition that allows them to, when they grow and matriculate as individuals, they're more resilient to the warmer water, which is what's driving the bleaching in Australia. Is that correct?
Yeah, that's it in a nutshell, basically. So because we've got two reasonably extreme examples of genotypes from two different environments, does that help in some way to try and figure out some of these questions around resilience, and can that then be fed back into restoration at some point in the future? So, there's never a question that we're going to be starting mixing corals from different locations of the world for restoration, but because it's such an extreme example, does it fine-tune what should be looked at a local level to then inform that restoration approach?
Awesome. Well, thank you very much, Jamie, and we'll catch you next time.
Talk soon, yeah. Cheers.